Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize winning lab. This channel has three main goals. One, to help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. Two, to build a supportive community where we can exchange the most valuable experience-based research advice. Three, to share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the third goal. Let's go! Let's make great science together.
Welcome to the Deep Dive. Today we're jumping into something pretty mind-bending in cancer research, a new way to tackle acute myeloid leukemia, AML.
Right, and this isn't your typical chemo approach.
No, not at all. It's about, re-educating the cancer cells, forcing them to mature, to stop being cancerous.
And the really wild part, the twist, is how they do it.
Exactly. They use a drug combo that suppresses a pathway, the WNT pathway, which, you'd normally think inhibiting its inhibitor would boost cancer. It's completely counterintuitive.
It really is. But it seems to work synergistically, offering a potentially less toxic, maybe more effective differentiation therapy.
Okay, so our mission today, unpack this Nature paper. Perturbing LSD1 and WNT rewires transcription to synergistically induce AML differentiation. We want to figure out how this works, why it's such a big deal, and, what it might mean down the road.
Sounds good. So let's quickly paint the picture of AML itself. It's a really tough blood cancer.
Devastating, yeah.
The blood cells, they basically get stuck. Immature, cancerous. They just don't differentiate into the healthy cells we need.
Yeah, the current treatments. Often pretty rough, right? Intensive chemo.
Very rough. Significant toxicity. And honestly, the long-term success isn't always great, so there's this huge, urgent need for something better, something maybe kinder.
Which brings us to this idea of differentiation therapy. I find this concept really elegant.
It is. Instead of just killing the leukemia cells, you try to, coax them along, get them to mature, grow up, and then die naturally, like normal cells do.
Sort of like convincing a rebellious teenager to finish school and get a job instead of,
Exactly. And we've seen it work brilliantly in one specific type, APL, acute promyelocytic leukemia, the ATRADO combo.
Huge success story there. But it hasn't really worked for other AML types, has it? That's been the roadblock.
Precisely. Which leads us to the key players in this paper. First up is LSD1.
Okay, LSD1, lysine-specific demethylase 1. What did that do?
Think of it like a brake on differentiation. It's an enzyme, a protein, that removes certain chemical tags from histones the spool's DNA wraps around.
Right.
By removing these tags, LSD1 helps keep maturation genes locked down, switched off. It traps the AML cells in that immature state.
So naturally, people tried LSD1 inhibitors, drugs, to release that brake.
They did, yeah. Showed some promise, but often ran into problems. Toxicity issues, limited effectiveness when used alone. Kind of hit a wall.
Okay, player one, LSD1. Who's player two?
That would be the WNT pathway, and specifically a kinase called GSK3.
Ah, the WNT pathway. Often implicated in cancer, keeping cells renewing themselves, right? Like an immortality switch.
You got it. And GSK3 glycogen synthase kinase 3 normally acts as a dampener on WNT. It tones it down.
Okay, so here's the weird part again. If you inhibit GSK3, wouldn't that activate WNT? And wouldn't activating WNT be bad in cancer?
That's the standard logic, absolutely. More WNT usually means more cancer fuel. But interestingly, inhibiting GSK3 has also been looked at directly for therapy. In some AML cells, weirdly, it can actually cause them to stop dividing and even differentiate a bit.
Huh. So it's complicated.
Very. And the problem was, drugs that just inhibit GSK3, like one called LY2090314, they didn't really do much on their own in the clinic. Limited success.
So you have LSD1 inhibitors, kind of promising but flawed. GSK3 inhibitors, also kind of flawed on their own. And with this confusing WNT link,
right,
which sets up the big question for this research.
Right.
What if you put them together? Could combining them unlock something powerful, overcome those limitations?
That was the core idea. Could they find synergy where the individual parts fell short? Especially given that confusing WNT signaling.
How do they even start looking for that synergy? Seems like a shot in the dark.
Well, they were systematic. They started in the lab trying to, as you said, re-educate cancer cells in a dish. They used a mouse model of AML, these ERHOXA9 cells.
Ones that glow green when they mature.
Exactly. Makes it easy to see differentiation happening. And they screened a whole library of bioactive molecules against these cells, looking for something that worked really well with a low dose of an LSD1 inhibitor.
They didn't want to use a high, potentially toxic dose of the LSD1 drug. Smart.
Right. Find a partner that boosts the effect.
And they found one.
They did. The top hit, the best synergizer, was that GSK3 inhibitor, LY20903114.
Wow. The very drug that didn't do much on its own.
Precisely. That was the pivotal moment, finding this partner drug that just dramatically amplified the LSD1 inhibitor's effect.
So what did that amplification look like in the lab?
It was pretty clear-cut, actually. The combo didn't just nudge the cells a bit. It robustly cranked up key markers of myeloid differentiation, things like CDL&B and GR1.
Molecular flags saying, I'm maturing.
Yep. And you saw way more of that green glow from the GFP reporter in the ER-HLXA9 cells.
Yeah.
Visually obvious maturation.
Okay, so they mature. But did it stop the cancer behavior? The immortality?
Crucially, yes. The combination synergistically slammed the brakes on proliferation, and it wiped out their ability to self-renew.
That self-renewal is the key to cancer's persistence, right?
Absolutely. In these tests, called colony formation assays, where you measure self-renewal, the combo-treated cells look totally different. They form these distinct diffuse colonies, a sign they'd lost their aggressive cancerous nature and matured.
And this wasn't just in the mouse model.
Nope. They tested it across a panel of different human AML cell lines, too. Found strong synergy there as well. Really knocked down their ability to form colonies. That's a big step towards human relevance.
Okay, this sounds promising. What about long-term effects? Did the cells just revert back once the drugs were gone?
That's what they checked with something called a serial replating assay. And the cool thing was, even after they washed the drugs away, the cells that had seen the combo treatment just progressively lost their ability to self-renew over time.
So it wasn't just temporary. It suggests a real, lasting reprogramming.
It really does seem like a more permanent shift in the cell's identity. And visually, using right gene sustaining, you could see these nice, mature-looking cells, not just, cellular debris.
That's incredible. So
Yeah.
the main takeaway here for you listening is this isn't just about killing cancer cells like chemo. It's fundamentally changing them, forcing them to grow up and ditch their immortality. Imagine convincing a cancer cell to retire.
A really different paradigm. Now, obviously, the next big questions were about specificity and whether this actually works in a living organism in vivo.
Right. Does it kill healthy cells too? That's always the worry.
A major concern. But they found this combo was remarkably selective. It hammered the leukemia cells but had minimal to no effect on normal, healthy bone marrow cells. Things like macrophages or those crucial hematopoietic stem cells were largely spared.
That's huge. Suggests potentially fewer side effects than standard therapies. A massive win.
Definitely. And they also checked if this was just a fluke with these specific two drugs.
Good point. Did it work with other similar drugs?
Yes. They tested other LSD1 inhibitors like bomedemstat and TK418, and another GSK3 inhibitor, 9-ING41, found similar synergistic effects.
Okay, so it's likely a class effect. The strategy of hitting both LSD1 and GSK3 works, not just those exact molecules. That gives options.
Exactly. More flexibility for development. Then came the big test. Mice.
Putting it to the test in a living system, how did it go?
They used a mouse model of AML, where the immune system is intact, a syngeneic model. The single drugs alone, they had maybe modest effects, slowed things down a tiny bit.
But the combination.
The combination was striking. It significantly reduced disease progression, leukemia burden, and, most importantly, it significantly extended the lifespan of the mice.
Wow, so a clear benefit in a living animal. That's a major hurdle cleared.
A huge leap, yeah. From the petri dish to showing real efficacy in vivo.
So for you listening, the key insight here is twofold. This combo seems targeted, sparing healthy cells, which is great news for potential side effects. And it works in animal models, which is a critical step towards potentially helping actual patients. It's looking like a smart bomb, not a carpet bomb.
Precisely. Which brings us to the really deep dive, the how, the molecular mechanism, and also the which patients might benefit.
Okay, let's get into the nuts and bolts. How does this combo actually rewire the cell?
What they found using techniques like RNA sequencing, which reads the cell's active genes, was profound. The combo didn't just tweak a few genes, it caused massive global changes in gene expression, far more than either drug alone.
A total reprogramming.
Basically, yes.
Okay.
It systematically cranked up the volume on genes needed for myeloid differentiation for maturing, and simultaneously, it shut down the genes associated with being a leukemia stem cell, the dangerous self-renewing ones.
And what about that WNT pathway, the counterintuitive part?
This is where it gets really fascinating. Remember, inhibiting GSK3 should boost WNT. But in the context of this combination, the opposite happened. They saw a clear suppression of the canonical WNT pathway activity.
It downregulated key WNT target genes.
Exactly. Even turned down a key WNT transcription factor called TCF1. It completely overrides the expected effect of GSK3 inhibition alone. It's like the LSD1 inhibitor changes the context entirely.
So the combo doesn't just add effects, it creates a totally new outcome. It reroutes the signals.
That's a great way to put it. And this rerouting led them to an unexpected hero pathway.
Which was?
The type I interferon pathway, IFN signaling. Turns out, LSD1 normally keeps this pathway somewhat suppressed in these cells.
Ah, so inhibiting LSD1 takes the brakes off interferon signaling.
Right. And when you also inhibit GSK3, this effect is massively amplified. You get this really strong activation of interferon pathway genes, and this pathway is known to push cells towards differentiation.
So releasing LSD1 suppression unleashes interferon, and the GSK3 inhibitor somehow turbocharges that.
That seems to be the core mechanism. And they drilled down further, identifying the key molecular players coordinating this.
The dynamic duo you mentioned earlier.
Yep. Beta-catenin, which gets stabilized when you inhibit GSK3, and IRF7, an interferon regulatory factor, which gets upregulated when you inhibit LSD1.
So one comes from the GSK3 side, one from the LSD1 side.
Precisely. And using advanced techniques like CT and RUN, which maps where proteins bind to DNA.
Like finding fingerprints on the cell's instruction manual.
Kind of, yeah. They saw a dramatic increase in beta-catenin and IRF7 binding together, co-localizing specifically at the promoters of genes in that type I interferon pathway.
So they physically team up at the control switches for these interferon genes.
It really looks that way. They're acting together, likely forming a complex to drive this differentiation program.
And was there one specific conductor this duo recruits?
STAT1 seems critical. It's a major transcription factor in the interferon pathway, known to be involved in differentiation. The combo synergistically activated STAT1.
And if you block STAT1?
Crucial experiment. Blocking STAT1, either with a drug or by genetically removing it, completely wiped out the differentiation effect of the combo.
Okay, so STAT1 is essential. The signal absolutely has to go through STAT1.
It appears so. And they even did experiments co-immunoprecipitation, showing that beta-catenin and IRF7 physically interact. They stick together, but only when both drugs, the combo, are present.
Wow, so they really only form this functional partnership under the influence of the combination therapy.
Exactly. And one more interesting layer. This beta-catenin IRF7 complex wasn't just activating genes, it was also found sitting on and downregulating key cell cycle genes like MYC.
The famous cancer gene MYC. So the complex can also act as a brake.
It seems context-dependent. It activates the differentiation genes but also helps suppress the proliferation genes, a dual action. They also clarified that most of the synergy, especially the beta-catenin part, comes from hitting GSK3 beta, though hitting GSK3 alpha also adds to the differentiation push.
Okay, that's a complex but really elegant mechanism. Rerouting WNT, unleashing interferon, driven by this beta-catenin IRF7 duo activating STAT1.
It hangs together quite nicely. Now, the next step was critical. Does this work in actual patient samples?
Right. Moving beyond cell lines to primary cells from people with AML.
They tested the combo ex vivo, so in the lab, but using fresh samples from 16 AML patients, and the results were encouraging. 11 out of the 16 samples responded really well, showing significant differentiation.
That's a pretty good hit rate. Did they see any patterns, like which patients responded best?
They did. They found genetic clues. The combo seemed particularly effective in AML cells that had specific mutations. MLL rearrangements or mutations in DNMT3A or NPM1.
Ah, so potential biomarkers already emerging.
Exactly. And interestingly, those DNMT3A mutant samples already had higher levels of certain immune-related signals, suggesting their interferon pathway might be sort of primed and ready to respond more strongly to the combo.
Fascinating. Any clues about resistance?
Yes, mutations in the TP53 gene, a common marker of difficult-to-treat cancers, were linked to resistance. That's also important information for clinical trial design. And again, they confirmed the combo was safe for normal human blood stem cells.
Okay, strong ex vivo results.
Yeah.
Especially in specific genetic subgroups. What about in vivo with patient cells?
That's where patient-derived xenografts, or PDX models, come in, implanting human AML cells into mice.
The ultimate preclinical test, really.
Pretty much. And in PDX models using AML with those DNMT3A mutations, the results were outstanding.
How good?
The combo significantly reduced the tumor burden, the amount of leukemia, and dramatically extended the survival of the mice. In some cases, they even saw complete clearance of the leukemia.
Complete clearance. Wow.
Really impressive outcomes in those models. They did note, though, that in models using AML without the DNMT3A mutation, the combo wasn't clearly better than just the LSD1 inhibitor alone.
Okay, so that reinforces the idea that DNMT3A mutation status could be a key biomarker for predicting who benefits most from the combination.
It strongly suggests that, yes. And they further validated this in complex 3D human bone marrow organoid models, adding another layer of confidence.
So bringing it all together for clinical relevance.
They looked at gene expression data from a large cohort of real AML patients. They calculated a synergy signature score based on the gene changes they saw with the combo in the lab.
And did that signature match up with what they expected in patients?
It did, beautifully. The synergy signature correlated positively with type I interferon signals and negatively with WNT signaling and leukemia stem cell signatures in the patient data. Powerful validation.
And the ultimate question, did that signature predict how patients actually did?
This was perhaps the most striking finding. Patients in that cohort with a high synergy signature score had significantly longer overall survival.
Incredible. So this signature, derived from the lab work, actually has prognostic value in real patients. It could potentially identify patients likely to have better outcomes or perhaps respond well to this type of therapy.
Exactly. It's a very strong link between the lab mechanism and clinical reality. And it hints this could be a valuable biomarker for future trials, plus the potential implications. This idea of suppressing WNT while rerouting transcription might not be limited to AML?
You mean it could work in other cancers?
Potentially, yes. Especially other cancers where WNT signaling is a known driver, like, say, colorectal cancer. It opens up broader possibilities.
Okay, let's pull back and look at the innovation here. What makes this work so groundbreaking?
Well, the core rationale was clever. Deliberately combining low-dose LSD1 inhibition with GSK3 inhibition, aiming for differentiation without adding toxicity. Targeting specific gene circuits.
It wasn't just randomly throwing drugs together.
Not at all. It was hypothesis-driven, and the way they figured it out was innovative, too.
The methods?
Yeah. High-throughput screening found the pair quickly, but then the multi-omics profiling RNA-seq, ATAC-seq, HETRUN that gave them this incredibly detailed map of the reprogramming.
Seeing how the chromatin landscape changed, how gene access shifted.
Precisely. They saw enhancer regions linked to self-renewal literally collapse, while differentiation gene regions became hyperactivated. It was like watching the cell's operating system get fundamentally rewritten.
And the results were just potent.
Extremely. Remember that tenfold reduction in the concentration needed for the GSK3 inhibitor and complete leukemia clearance in some animal models with minimal side effects on normal marrow. That's remarkable efficacy.
It also sounds like the research ecosystem played a role.
Definitely. The paper highlights collaboration between basic science labs, clinicians, and biotech. That kind of translational network is crucial for moving things forward quickly.
So positioning this breakthrough, what's the significance?
I think it really revitalizes differentiation therapy for AML beyond the niche success in APL. It shows it can be a powerful strategy more broadly.
And it's close to the clinic.
That's a key point. Both drug classes, LSD1 inhibitors and GSK3 inhibitors, already have drugs that have been through phase I safety trials in humans.
So they know the basic safety profile.
Right, which means this combination is, as the paper says, readily testable in clinical trials. It significantly shortens the timeline compared to developing entirely new drugs.
Plus, the biomarker angle seems really strong.
Absolutely. The link to DNMT3A and NPM1 mutations and that synergy signature points directly towards biomarker-guided trials. Personalized medicine in action, identifying the patients most likely to benefit.
It really feels like a prime example of dual pathway rewiring, hitting two connected pathways to achieve an outcome neither could alone overcoming resistance, minimizing side effects.
That concept of fundamentally altering the cell's identity by manipulating its internal wiring, it's incredibly powerful.
So this deep dive really highlights an amazing strategy, not just killing cancer, but truly rewiring it, forcing it to abandon its cancerous identity. The counterintuitive nature of it, how inhibiting pathways in combination leads to this unexpected beneficial reprogramming via interferon signaling, is just fascinating.
It really challenges us to think differently about drug combinations and cellular plasticity.
Which leads to the final thought for you listening. We've seen how targeting LSD1 and WNT can rewire AML cells. But what other pathways, what other cellular circuits could we learn to manipulate? What other seemingly contradictory combinations might unlock completely new ways to re-educate cancer cells, tricking them into giving up the fight across different types of cancer? That feels like the next frontier.